In this video, you guys, I would like to talk about translation. Last week we started with transcription, and this diagram told you last week that it's a really busy diagram. There's a lot of stuff going on, but we made sense of this portion of it. So here's our DNA. There we see the unzipping. And right here, we see messenger RNA. Remember, messenger RNA, mRNA. This is the copy of the DNA that's safe to leave the nucleus. The DNA itself never leaves the nucleus. It's just too important for that. And so now that messenger RNA is the end result of transcription. We've got a copy of a gene, and now that copy has to leave. It has to go out into the cytoplasm. And now we're going to make sense of this part of it. That messenger RNA is going to get translated. Just like any code, anything written in code, at some point in time you eventually have to translate it. And so that's what we're going to look at. And the translating actually is going to create our end result is a protein. That's the end result of our translation. So again, just a quick review, what we looked at last week. Replication, transcription, location is important. Those both occur in the nucleus. And then we had that messenger RNA that leaves the nucleus, and translation occurs out in the cytoplasm. And the idea is if something would happen to that messenger RNA, if it would get harmed, destroyed, whatever happens, you can always go back into the nucleus, and that DNA is safely in the nucleus, and so you can keep making as many copies as you want without that original DNA ever getting harmed. And so that messenger RNA is kind of a one-time use, and then it disintegrates and it goes back, and then the pieces actually get made into a new strand of messenger RNA for a different protein. Three types of RNA, remember, we'll get to that in a second. No T's, right here, no thymine, instead it has uracil. And so DNA is, but in RNA, we will still see A's, but we won't see any T's. We won't see any thymine. So some big differences there. DNA is double-stranded. RNA is single-stranded. So there's a number of differences between the two. The sugar that's present, ribose, it's different between DNA and RNA. So there are some noted differences. Biggest thing are the three different types. And for transcription, messenger RNA, we saw this in transcription. And we'll also see this in translation. Ribosomal RNA we'll see today. That's in translation only. And transfer RNA is in translation as well, not in transcription. So mentioned all three of them, but with transcription, we only saw messenger RNA. Today we'll see the other two in action, so we'll actually see all three. They'll all three be working together. Here's our transcription complex. We talked about that. Messenger RNA, or RNA polymerase, and the other one I'd like you to remember is helicase. Helicase does the unzipping. So there's that transcription complex. We see the DNA getting unzipped. And RNA polymerase starts to bond new bases. Remember, A's use C's and G's. And so we see that growing strand of messenger RNA as that complex is moving along the strand of DNA. It's unzipping on one end and re-zipping on the other end. And so just some differences in terms of what we've seen so far. Once the gene is done, this is now the completed transcribed gene. That transcription complex breaks up. These pieces were the transcription complex, that series of enzymes. They break up, the DNA rezips, and now this messenger RNA leaves the nucleus. And so that's kind of the end of transcription. The goal there is just to get a copy of a gene that we can safely take out of the nucleus. We looked at this picture as well. We saw the base pairings. G still bonds with C, C still bonds with G. A T would still give us an A, but here is the big difference, that A bonds with the U, not a T. So remember, in RNA, we don't see any T's, we see U's instead. So now we get to translation. So now we've got that messenger strand, and we're going to try to make some sense out of it. So that messenger RNA copy is used to assemble amino acids. Remember, we talked about amino acids a long time ago in our macromolecule unit. There's 22 different amino acids, and we're going to see those in this unit and utilize those to make proteins. The ultimate goal is to make the protein. Every 3 nucleotides in the messenger RNA specifies a particular amino acid. That's called a codon. 3 nucleotides, and we'll see this as well. That's called a codon. C-O-D-O-N. So that sequence of 3 has a name. And oh, there it is right there. It's called a codon. And then the opposite, anti means against or opposite, transfer RNA has the anti or the opposite codon. And we'll start to see that a little bit in action today. So it'll hopefully start to make some sense. And then the ribosomes. That's the rRNA, and that's the job to pair the codons with the anticodon. So here on this slide, we see them all in action. We see messenger RNA, transfer RNA, and rRNA will all be in action, all 3 varieties, in order to get that protein constructed. So here it is. It's a little confusing. So here the green, it almost looks like a hamburger bun in my opinion. The ribosome or the rRNA. That's the green portion. Here we see the messenger RNA that was made in the nucleus, remember, and it was allowed to leave. And this big blocky thing right here, that's the transfer RNA. And it's called transfer RNAs because it's a transferring the amino acids. And up here, I'm just going to abbreviate amino acids like this. There's an amino acid. There's an amino acid. And so there we see the amino acids being transferred in. And so we see a start codon, AUG, and then we see the anticodon. Anti or opposite. Opposite of A is U, opposite of U is A, and opposite of G is C. And so transfer RNA has what's called that anticodon. And then here, peptide bonds start forming in order to create those amino acid chains and that ultimately that protein. And so now what happens is this transfer RNA is going to leave. This one's going to slide over one space. And then in the next slide, we're going to see a new transfer RNA coming in to bring in the next amino acid. So the transfer RNA with the amino acid attached to it binds with the messenger RNA inside the ribosome. And then that bond is made, and then another transfer RNA comes in, bonding next to the original transfer RNA, and those amino acids from the transfer RNAs bind to each other, drop the transfer RNA, and this is the beginning of that polypeptide chain. Or the start of what we would call a protein. And so that polypeptide chain is the beginnings of the protein. And so now this one, notice it dropped. Its amino acid is right there, so it's leaving. This one's waiting for the next one. In comes the next one. And we have codons and anticodons. And that process continues. So now this amino acid, this CYS, eventually will get joined to this. And then here we see that growing protein. And so in terms of codons, and we'll see these more in a moment, there's actually a codon that signifies start, as in begin decoding here. And there is actually a codon that signifies stop, meaning the protein's done, stop coding here. So there is actually 3 letter codes for starting and stopping. And last piece, this animation right here is computerized. It's not an actual cell, but it's computerized, showing what's actually occurring in the shell. And the cool thing about this animation is it's showing the process in real-time speed. And you'll see that this is not a slow process. It's actually crazy fast. You'll hear him talk about things are spinning at the speed of a jet engine. And so we'll get to the idea of a mutation down the road. But as you watch that animation, think about how fast it's going and the possibilities for mistakes, because they do happen in this process. Mistakes